right, please be seated. The state may call its next witness.
This time the state calls Eric Koeppen.
Okay, good afternoon. Please stand in back of the witness chair. Raise your right hand. Do you swear under penalty of perjury that you will truthfully testify?
I do.
Please have a seat. Remove your mask. And then state your full name and spell it.
My name is Eric Koeppen, and last name is spelled Koeppen.
Okay, a couple of things to keep in mind when you're testifying. Yes or no answers because of the court reporter. No or nodding your head.
Sounds good. Thank you.
If there's an objection, stop talking immediately and don't answer unless I tell you to. If you don't understand a question, let the lawyer know. And two people can't be talking at the same time. So wait until the question's finished before you answer.
Okay.
All right. Counsel?
Mr. Koeppen, could you state your occupation for the record?
I work as a forensic scientist in the biology section of the BCA laboratory.
How long have you been employed in the biology section of the BCA?
I've been employed there since February 2017.
About 4.5 years then?
That is correct.
And what are your current duties in the biology section?
As a forensic scientist, we examine items of evidence, process that item of evidence for any sort of biological fluid, do examinations, take notes on that item, make any collections. We'll then process that item for DNA profiling, make any comparisons to known samples provided in that case, write up a report, do reviews on other scientists' work, and then testify in court as needed.
What is your educational background to do such things?
In 2013, I graduated from Hamlin University with a Bachelor's of Science in Biology. I also graduated with a Forensic Science Certificate from Hamlin in 2013 as well.
Have you received any specialized training since your employment at the BCA?
Yes. Upon my employment at the BCA, we are placed into a training program. This takes about a year. You are expected to observe other scientists in the laboratory, attend classroom lectures, perform written and oral examinations, and then complete a competency test at the end of our training.
And are you evaluated after that competency test? Are you evaluated regularly to verify your proficiency in conducting your work?
Yes, we take what are called proficiency tests. Those are twice a year. They're used to kind of examine our workflow and make sure that we get the correct results of the test.
And you've testified as an expert before, correct?
That is correct.
All right. Now, before we talk about the DNA testing you performed in this case, would you please explain to the jury quickly what DNA is?
So DNA is a chemical or a molecule that's found within the cells. It kind of is the makeup of who we are as a person. Gives you your hair color, your eye color, things of that nature. It's highly unique to certain individuals besides twins.
Is it possible to identify, besides twins, the DNA of a particular person?
Yes, it is.
When you identify the DNA of a particular person, do you have a name for that result?
We would call that a match.
A match. Oh, I'm sorry, but just the actual, what's it called, DNA profile? Is that of an individual? Is that what that's called?
At the end of our testing, we do obtain, yes, a DNA profile.
Profile of a specific individual, for example.
Of an individual, or generally after the end of our testing, it's an unknown profile until we make our comparisons.
Okay. Now, is it possible then to distinguish the DNA profile of one person from the DNA profile of another person?
That is correct.
Okay. Now, is it possible to compare the DNA of a known donor with the DNA found in an unknown sample containing DNA to determine whether that person's DNA matches the DNA profile found in the sample?
Yes, sir.
Is that sort of the middle of the road, what you do on a daily basis, sir?
Yes, I could agree with that.
What is a single source DNA profile?
So a single source profile is one that we determine to be coming from a single individual. We determine this since you get half of your DNA from your mother and half of your DNA from your father. At most, at the locations that we're testing, we would expect 2 types. If there are more than 2 types at the location, then we can call that a mixture of DNA.
So when you do have 2 single source DNA profiles, given the unique nature of them, are you able to determine if those two profiles are a match or not a match?
Yes.
Okay. What other types of DNA results do you expect to find besides a single source result?
Like I stated earlier, we do end up getting mixtures of DNA where there's multiple contributors to that sample. There are often times where we get samples that we're unable to make any sort of identification to.
Is testing a sample of blood recovered from a scene something you do on a regular basis?
Yes, it is.
Are you typically able to recover DNA from swabbing the blood sample?
Yes.
And when you test blood samples, are they typically single source profiles?
Yes.
But they could be mixtures.
That is correct.
All right. And you'll be able to determine that whether it's a single source or a mixture based on the DNA profile you receive, correct?
Correct. During our interpretation is when we'd be making that determination if it's a single source or a mixture.
Okay, I just want to move now on to the testing that you perform at the BCA. Based on your training and experience, do you have an opinion as to whether or not the DNA testing methods used at the BCA are generally accepted in the scientific community?
Yes, they are.
Does the BCA have a particular protocol or methodology that is used when performing DNA testing?
Yes.
Okay. And can you explain briefly the DNA testing policy?
Yes.
Or process, I should say.
Process, sure. DNA profiling is done generally into 4 steps. The first step is extraction. We'll get a sample subjected to certain chemicals to help break up the cellular material and release that DNA into a solution. We'll then purify that solution to get that extract of DNA, so that purified DNA. This will bring us to our next step, which is quantitation. We'll see how much DNA is present within that extract and see if it's suitable to move forward in the profiling process. If it does meet a certain threshold, it moves forward to the next step, which is called amplification. We'll be targeting certain areas of that DNA for identification purposes, and we'll be focusing on those areas, making millions upon millions of copies of that area or those areas. And then once that is completed, the final step is interpretation, where that basically that sample is injected into our instrumentation. A profile is then generated. We'll then basically visualize that profile and make our interpretations.
Okay. Now I have a couple of questions about items which are collected and inventoried at the BCA. When an item is collected, are they typically given both a BCA lab item number and a description of what the item actually is?
Yes, when items are submitted, they're given a BCA lab item number, as well as a description that's provided by the agency.
So if you take an item out of the vault, you're going to have an opportunity to see both the item's lab number and then that description of the item.
Generally, the description is not written on the item itself. However, it is in our laboratory information management system, so I'll be able to see that on the computer, yes.
Okay. And those numbers and descriptions typically would be provided by the scientist or agent who collected or inventoried those items, correct?
That is correct.
All right. And those numbers and descriptions, do you utilize those when you issue a report about your findings?
Yes.
Okay. And that's done obviously to aid anyone reading it?
That is correct.
All right. Now I want to move on to the testing you did in this case. Did you generate a report documenting the testing that you performed?
Yes, I did.
Were certain items of evidence in connection with this investigation inventoried at the lab?
Yes.
Did that include a blood stain or sample from a known individual named Daunte Wright?
Yes.
And that blood stain was identified as BCA item 30?
That is correct.
Were there also a series of swabs taken from blood droplets located in this case?
Yes, sir.
And of the items, would you agree that based on the item number and the description provided to you, they are the following? Item 22, that's blood from the vehicle's driver's seat. Item 25, blood from the vehicle's front passenger dashboard. Item 26, blood from a vehicle's passenger side headliner. And then item 27, blood from the vehicle's rear driver's seat.
Yes.
All right. So you obtained those swabs to conduct testing on them.
That is correct.
And did the swab, the known swab of Daunte Wright, give you a full DNA profile?
Yes, it did.
And were you able to obtain separate DNA profiles from those 4 swabs of blood?
Yes.
Okay. And of those 4 DNA profiles, am I correct that each one was a single source DNA profile?
Yes, you are correct.
All right. Did you compare the DNA profile from each blood sample to the DNA profile of Daunte Wright?
Yes, I did.
Okay. And Are you prepared here to report the result for each?
Yes.
All right. Let's start with the first one, item 22, blood from the driver's seat. What was the result of your testing?
So blood from the driver's seat, item 22, was a single source male DNA profile. That male profile matched Mr. Wright's, and that profile would not be expected to occur more than once among unrelated individuals in the world population.
Okay. Next, item 25, that's the blood from the vehicle's front passenger dashboard. What was your result there?
So from the front vehicle dashboard or passenger side, it was a single source profile. It was an unidentified female profile. It did not match Mr. Wright's, and there were no further comparisons on that profile.
Were you provided any DNA samples from any other known individuals?
No, I was not.
So you had no DNA profile from an individual named Delana Albrecht Payton?
No, I was not.
Okay. Item 26, blood found on the the passenger side headliner.
Item 26 was also a single source male DNA profile. That single source profile matched Mr. Wright, and once again, that profile is not expected to occur more than once among unrelated individuals in the world population.
And then item 27, blood from a vehicle's rear driver's seat.
Item 27 was a single source male DNA profile, matched Mr. Wright, would not be expected to occur more than once among unrelated individuals in the world population.
Okay. I'm now going to show you a series of photographs already admitted as evidence in this case. Could you please show Exhibit 191? I'm showing you Exhibit 191. Do you see the sticker on there which states 22?
Yes, I do.
Okay. Could you go to the next Exhibit 192? And you'd agree that there's a sticker on the front seat there that says 22?
Yes.
All right. And based on your training and experience, to what would you conclude that this 22 corresponds?
This number 22 corresponds to item 22, which I previously reported.
Okay. And that report was that it was a match of Daunte Wright's DNA profile?
That is correct.
Okay. Next, please show Exhibit 193. And then move on to Exhibit 194. Would you agree that Exhibit 194 shows an item 25?
Yes, sir.
Based on your training experience, would you assume that this corresponds to the item number 25 that you tested?
Yes.
And this was the DNA profile that was a match for an unidentified female?
It was an unidentified female profile.
Okay. Next, Exhibit 195. And then, can you see the item 26 on the roof of that vehicle, sir?
Yes, I can.
Okay, and then go to Exhibit 196. And based on your training experience, do you believe that this was the lab item number you tested, that was number 26?
Yes.
And am I right that the result of your testing showed that blood was also a match for Daunte Wright's DNA profile?
That is correct.
Okay. And then finally, please show Exhibit 197. And then move to 198. I'm showing you an item, if you could zoom in on that, Mr. Miller. I'm showing you Exhibit 198. Would you agree, if we close up there, that it shows item number 27?
Yes.
And based on your training experience, are you to believe that that is a match for that is the blood sample that you tested as lab item number 27?
Yes.
And that, too, was a match for Daunte Wright's DNA profile? Correct?
That is correct.
All right. You can take that down. So to summarize your findings, of those 4 blood items, 3 of them were a match for Daunte Wright, and that 4th one on the passenger dashboard was from an unknown female DNA profile. Right?
That is correct.
Okay. No further questions.
You may step down. The state may call its next witness.
This time the state calls Travis Milan.